Lecture 9.
Okay. I assume that you all saw your grades posted, but I'll go through a discussion of the results at the end of the class when most of you are here. So I'll save the comments about the exam until there, although this generally you did very well in the class. But we'll address that more specifically at the end. The subject for today is molecular-based contraction-induced muscle tissue and its physiology. And there will be some hypothetical questions if you want to get your clickers out, because they'll be scattered through the lecture. Just some, general understanding questions. You won't be graded on whether they're correct or not, just on participation. So anyway, I'll have some participation points, but it's mainly so you can see if you understand things as I go through. Okay, we already introduced when we're going through histology some general comments about muscle, what it is, and it's classed into groups and so forth. But just for review, once again, this is the type of tissue that has the ability to shorten or contract. And it's divided into three major groups. Skeletal muscle is the muscle that attaches to muscle and allows us to move around our environment and so forth. It's striated, which is a histological observation, and we'll discuss why it occurs and learn the nomenclature for that in this lecture. We classify skeletal muscle contraction as voluntary, although in the same category as reflex contractions, which are skeletal and respond to stimuli such as pain. So voluntary has a broad description here, but we'll get into that later more. Smooth muscle is the muscle that provides the motility for the gastrointestinal system as well as reproduction, and our bladders, both gallbladder and urinary bladder, are controlled or moved by smooth muscle, as well as most of the vasculature, most of the blood vessels. It's non-striated and generally classed as involuntary. And cardiac muscle, only found in the heart, restricted to the heart itself. It's classified generally as involuntary, and it's striated and has much of the same striatic appearance as does skeletal muscle. And finally, there are, almost all cells have some contractile fibers that are used for such things as the division of cells and mitosis and so forth, the processes that even phagocytosis and diapedesis that single cells undergo, often facilitated by single contractile fibers within cells. So what we're talking about is generally applicable to all skeletal muscles, but we use as the prototype for this lecture skeletal muscle, and then we move on mostly in second semester to talk in detail about the heart. Our smooth muscle will talk about how these two classes more specifically differ in mechanics and structure and control from skeletal muscle. Okay, this figure represents a skeletal muscle cell, which we call a fiber. The term is synonymous. Skeletal muscle fiber is the same thing as a cell. These cells are surrounded by a plasma membrane, just like all of our cells, but for some silly reason we call it a different name. Sarco is the term which we refer to with muscle a lot clinically, and so sarcolemma is the membrane that surrounds or encircles a muscle fiber. As I've already said, skeletal muscle cells are distinct in that they're multinucleated, and you see that represented in this micrograph, and you see they're clustered along the edge just underneath the sarcolemma of the fiber. Most of the sarcoplasm within a skeletal muscle fiber is composed of contractile filaments, and they're divided into different clusters. So the individual contractile filaments we call myofilaments as a family, and we're going to subclassify them into different groups in a second. And the myofilaments are held within bundles called myofibrils. So composed of myofilaments within myofibrils. So myofibrils, I don't know if I said that right, but myofibrils are bundles of myofilaments. Sarcomere is the term for, I said sarco was muscle and mere is a fragment. So it's a fragment of a contractile cell. It's the part of a muscle fiber that can facilitate shortening, and we have divided it into marks so we can discuss its function, and originally to learn how it works. So we call a single contractile unit a sarcomere, and extends in this diagram between two Z lines. Sarcomeres are organized in muscle fibers in series, so one follows another into a long series of sarcomeres, and so their contractile effect is multiplied by this series architecture. The striations which we see represented here are due to the effect of the myofibril organization, which we call register on the conductance of light through the tissue. And so you can see a sarcomere here extending between these two lines in the middle of clear spaces, which we call Z lines. So a sarcomere is one unit of that long length. The thin filaments will refer to two families of filaments we call myofilaments, one thick and one thin. And the thin filaments are indicated in this artistic representation as green. So the thin filaments are attached to the Z line, and the thick filaments, as you see represented in orange here, are located between the sets of thin filaments. We also describe or discuss the sarcomere in terms of the striations themselves, whether they're dark or light. The original designation was I-band and A-band, and I stood for isotropic and A for anisotropic. Your book's actually wrong, but that's expected, because for many years, one of those things, you get something in publication, the next author just copies it, and then the next, and so forth. So what it says in Bourne and Bullpape is isotropic is due to the conductance of polarized light through a muscle section that makes it look clear. It's actually, if you send polarized light through the muscle, a slide of muscle, it will look dark where the I-band is and light where the A-band is, and the phase contrast actually matches, phase contrast microscopy matches this type of figure. Well, you see that darker A-band for anisotropic and the I-band for clear. Isotropic means conducts light evenly, and indicates also something about more intensive light, where anisotropic is less intense light because it's getting interfered with. But anyway, that's where the names originally came from. The Z line also appears dark at each end of the sarcomere, and the H band is a lighter area which bisects the A band. Once again, I said that the thin filament is the green area on this depiction, and the thick filament is the orange, and so the boundary of the thick filament is the same width as the A band, as you can see. It's also, to really understand how this works, you have to have a figure in your mind, kind of a picture of what a cross-section. This is all two-dimensional, and that really confuses us. And so if we cross-section this two-dimensional representation, you can get an understanding of the orientation of the fibers. So the one cross-section, which occurs in the middle of the I band right here, just next sort of the Z line, you'll see that the thin filaments are actually organized into a hexagonal or a six array of six as represented here. And the thick filaments are, if you cut through this A band right here, you'll find that they're in the center of each of these arrays of six. So there will be thick, here's one example, this thick filaments are out by six thin filaments or actin molecules. And if you cut through the center of the H band, you'll see that they're the array of the thick filaments without the actin surrounding this. And this organization's important, as I mentioned before, because of the way myosin interacts with actin, as we'll see in just a second. So this is a nice summary of what we've been talking about as far as how a muscle, skeletal muscle, is composed. This is a deltoid muscle, and actually the muscle fibers themselves are bundled in two bundles surrounded by a membrane, and each bundle is called a muscle fasciculus, but we'll get into this in the next lecture. Within the fasciculus are muscle fibers, and then within the muscle fibers are myofibrils, and within them are myofilaments, and then this reminds us of the bands. The A band is where the thick filaments, and the I band only has thin filaments in the Z line. So now to understand even more about how molecular mechanism works, we look at each individual fiber separately, and we'll start with the thin filament. And the green part in this figure that's represented looks like a beaded necklace. We always use that example. If you had a necklace of pearls and put two strands and twisted them, that's what it would look like. It's like two strands in we call it an alpha helix of beads, and you can see that represented by the two green columns of beads. Each of those beads, if you will, is a globular protein called G-actin. So actin is actually a globular protein, but in a solution of the same ionic strength as body fluids, they polymerize automatically. It just comes together into a polymer that's called F-actin. And so anyway, this is just a spontaneous polymerization process. For the G-actin forms F-actin in solution, get to a certain concentration. So the F-actin is a combination of individual globular actins. And the double helix itself we call F-actin for fibrous actin. That's not the whole thin filament story. The thin filament is composed of several types of proteins. The second one is represented in brown in the figure, and it's a filament protein, but this filament protein is also present as two identical alpha helices. And so the brown here, that's almost, we usually say near the groove of the actin, F-actin, is tropomyosin. And these tropomyosins are laid end to end. In other words, they're not long. It's not a long protein that extends the whole length of the F-actin. It extends approximately 7 beads or 7 G-actins. So each strand of tropomyosin, double strand, extends about 7 actin molecules, then the other one is right against it, another 7. The alpha helix goes all the way around 360 degrees with about 13, 14 actins. So one tropomyosin is about half of a revolution of the actin, F-actin. And there's two, as you can see, two tropomyosins on each side of this actin. So each groove has a tropomyosin located near it. Troponin is a third part of the thin filament, and it itself is a trimeric, a heteromeric protein, a complex protein. It's globular in nature, but it has 3 different major parts, as we're going to mention. Tropomyosin T, or TNT, is represented by this purple sphere, and it's named because it binds to tropomyosin. So that it's the part of this globular protein that couples to tropomyosin. You'll notice that there's one troponin for each tropomyosin. So remember, there's 7, they extend 7 lengths. So that for every 7 lengths of active beads, there's one tropomyosin coupled to it. One troponin coupled to a tropomyosin. TNC is the second component of the trimeric troponin, and tropomyosin C was named because it binds to calcium, and you can see it represented in yellow in this little figure, or just figure. It's like many calcium chelated proteins, has a couple binding sites for calcium. It actually has a potential to bind to 4 calciums, and 2 of those sites are called high affinity and 2 low affinity. The ones with high affinity are almost always bound to calcium, and the lower affinity hook up and unhook to calcium as calcium concentration changes, which helps regulate the whole process, as you'll see. The third component of troponin is troponin I, and it's pictured here in kind of an orange color, and it's named because it inhibits interaction of myosin and actin. It blocks the actin binding site, which is on each actin, G-actin, for myosin. So the binding site is shown right here in white, and it's masked at this point by TNI, troponin I. And so that kind of starts indicating how we control the process, as you'll see. Okay, so let's talk about the other filament before we try and get through how contraction occurs. The other filament, the thick filament, is not near so complex. It's just made of myosin, a contractile protein called myosin 2. It turns out there's several different forms of myosin, so this one we call myosin 2. And it's in a column. The myosin 2 molecules are in a column that we call bipolar, which means that the N-terminals point out, the N-terminal ends of these proteins point out each end with a C-terminal in the center. So it's called bipolar. Any given column or cluster of these myosin 2s is called bipolar. The myosin is composed of what we call heavy chains of myosin, and they're first coupled into a tail region, which is interwoven, is alpha helical, and then a hinge region. So the same long myosin forms this hinge that has a little turn, and this comes up from the column, and then it spreads into forming at the N-terminal end of the myosin 2 globular heads. So you have the tail that coalesces to form the cluster of myosin or rod. Then you have the hinge that points outward away from the rod, and then the N-terminal is a coiled kind of a globular protein. Finally, the globular protein associates with 2 we call light myosin chains, or light chains, and there are 2 different sorts we found out not too long ago. One's called an alkali light chain, and the other's called a regulatory light chain. And the alkali light chain in this picture, they've just, for sake of discussion, colored kind of the aqua color. And that seems to hold the head into this stable globular form, so it's mainly there to maintain changing the myosin, the end of the myosin into a globular array. The other light chain in red is what regulates the activity of myosin ATPase. And we'll talk about that as we move along. But the heads do have binding sites for both ATP and for actins we've talked about. So each of these globular heads can bind to two different sides, one to an ATP and one to, as we've already pointed out, the part of the thin filament actin that has a binding site for it. There are some accessory proteins that facilitate the holding and structure of this, not so much the contractile activity, but hold us in this register array. Titin, T-I-T-I-N, is named because it's the largest protein we're so far aware of. It's a huge protein in terms of how many amino acids it's comprised of. But it's tethered to myosin and also to both the Z lines and the M line in the center. So what it's doing is it's holding that thick filament in the center space. Some people, in some of the pictures they show it as a big spring because it has elasticity, and we'll talk about that when we get to the mechanics part of the story. But we talk about passive or resting elasticity. If you have the muscle relaxed and you pull on the muscle, it resists that. It has elasticity just like the rubber band does, and that is somewhat, among other things, a product of this myosin's form. So it's kind of holding the thick filament in the middle of the array, and also holding the sarcomere width to the given width, if you will. And finally, there's nebulin that we don't know as much about. It's represented as this kind of red, I guess, spiral that goes around the actin. Some people call it a molecular ruler. And if you take cell biology, you'll learn that actin, I said it spontaneously, G-actin spontaneously forms into F-actin. Well, you need something to stop this occurring in the cell. Like actin strands keep forming and falling apart, forming and falling apart continually, and part of the cytoskeletal process. Nebulin stops this and holds that actin to a given length. So that's what we know about it right now. And again, the bipolar nature of the thick filament is shown in more detail later. So heads are oriented towards the distal end of the column, and there's a bare zone, just the rod part of the array in the center, where that M line is. So, and the other part of this story is important. So the heads come out from the column in 60-degree angles, so they come out, 6 of them come out all the way around the 360 circle of the filament, if you will, and hence they have the ability to, there's a head that's available to couple with every actin. Remember, there's a cluster of 6 actins around the myosin. Okay. So now we're finally ready with understanding, kind of basic understanding of the architecture of skeletal muscle. We'll talk about how it works. And for many years, and I think we still call it the sliding filament hypothesis in many texts, but we can, I think, we certainly know it's not a hypothesis anymore. So we're going to talk about what we mean by the sliding filament mechanism of contraction, and then the molecular mechanism that allows that to occur. This was understood with actual photography years before we knew molecularly how it did. And we can isolate muscle filaments out of muscle and lay them in a petri dish or on a slide and cause them to relax or contract. It's not a very involved thing. We do it in some undergraduate labs very simply. So anyway, this represents a resting individual filament, and then this is one that's intermediate, and finally this is one that's contracted. And so as you see, what happens when it contracts is the sarcomere width decreases as the filaments slide past each other. And you notice the A band doesn't particularly change in length. None of the filaments shorten, so what's going on is that the filaments are sliding past each other, which leads to a very typical exam question on how the sliding filament hypothesis works. So if you have your eye clicker, just for your entertainment, what do you think the right answer is? Contraction of skeletal muscle, does the A band width decrease to the filament shortens? What would be a good indication that the width is changed? I'll go about 30 seconds. So once again, this is just participation. I'm not going to grade you on your answers because you just heard this story. So just to help us keep your mind kind of on the same thing. So I'm ad-libbing 138. Okay, a few more seconds. 5, 4, 3, 2, 1. Okay. And at least 80% of you got the story. So each sarcomere width is decreasing. If you multiply that times a linear array of these, you get a significant shortening of the entire muscle. And the way it shortens, of course, is that the filaments slide past each other. So if this is the resting configuration, you'll notice that the actin slide past the myosin. In fact, this is true and becomes a significant part of the story when we get to the mechanics. You notice the actin actually overlap. And if you're thinking about what's going on, you realize that will affect the strength of contraction. And we'll talk about that a little bit later. Okay, what's the mechanism that allows these contracted filaments to slide past each other? We call it the cross-bridge cycle, and we'll go through it step by step. But what's going to happen, of course, is the actin and myosin filaments are going to slide past each other. So we're going to start out right here. So we're going to start out with what we call the attached state. And right at that point, it's capable for ATP to bind to myosin. So this right here has no ATP. ATP binds, I show you a little ATP with the 3 phosphates hanging from it. And once that binds, the myosin head switches to what we call a low affinity state. So the binding of ATP to the myosin head disrupts the bond between actin and myosin. There's no more affinity of the myosin head to the binding side of actin. And so the dissociation occurs. So that occurs right here. The next step is that ATP is hydrolyzed. Right here it's coupled to 3 phosphates. So remember the ATPase is part of the myosin head itself. And so immediately the ATP is hydrolyzed, which is right here. And once it's hydrolyzed, it changes a head conformation. The myosin head actually goes closer, they usually say from 60 degrees to 90 degrees. So it goes more upright, as you can see, going from here to here. And that moves you down something like 2 beads or so, a couple of nanometers down the strand. So just the head turning up moves the myosin orientation with the next binding side of actin further down the actin. And also, it shifts the head to what we call a high affinity state, where it wants to bind again to actin. And so then it binds again. So in other words, once you're hydrolyzed to ADP, now and in that cocked state, I tried to not say the word cocked very much because it might get you excited. I'll try not to say cocked too much. So anyway, on this cocked state, which you see right here, the cocked state has a high affinity, so the thing's right up there ready to go, as in cocked. And in that particular situation, the myosin couples to the actin that you can see right here. As soon as that happens, that allows, evidently it's like a spring-loaded machine because the binding itself causes the head to go back to the relaxed conformation. So you see the difference between here and here is the head goes back to the 60-degree conformation and phosphorus is released. So evidently this is releasing potential energy or stored energy in the position of the head, and so it goes back to the relaxed state. And when it does, that causes the myosin to move by the actin. So that's called power stroke. So the change in head conformation, remember the head is still bound to the actin, pulls the myosin past the actin, and so that's called the power stroke. And then the ADP is released, adenosine diphosphate is released after it shifts to that low energy state, and that once that's released, that provides a binding site for a new ATP to couple, and so then we start going around it again. So it keeps flipping back and forth. And remember, there's 6 of these all around the column, or 6 circularly around the column, all of these going really, really fast, and so this cycling continues and continues as the actin pulls by the myosin. So the question that's probably not evident as you went through this, what would you expect if you ran out of ATP? This is an extremely ATP-consumptive process. For every cross-bridge cycle, 1 ATP is burned up. And muscles don't store a lot of ATP. They have to continually produce it. So what would happen if you ran out of ATP, just as a first guess here? And again, the answers aren't important. I'm just trying to get you to follow along. Some of you have had some of this before, and so some of it's more obvious to some of you than others. Okay, about 5-6 seconds. We had 138 last time. That's probably about it. 2, 1. Somebody that was asleep last time woke up. Okay. Oh, a lot of differences in response. Why is it doing that? It's contracting. Oh, it's still collecting data. Oh, cool. We'll go up to 140. I couldn't figure out what you kept saying. Stop. Anyway, letter C, you really, unless you had another class, you know a lot about the subject, you probably couldn't answer now, but we haven't even talked about polarization of the membranes, and that's coming this unit. We'll talk about membrane potential and how they occur and action potentials. That's all in this next couple of weeks, so we got some heavy material. But I'll tell you that it doesn't, when you run out of ATP, it doesn't immediately depolarize. So, but you wouldn't know that unless you were smart like I am. So, and you're laughing appropriately. My wife would join you. So, the real answer is B. You'll be locked in tetany, and that is, let me go to the next slide if I can get the thing to work. That's right here. So here's another cross-bridge cycle by another author, and the little story about running out of ATP is right here. So it's the same story. If you have the ATP bound, it's just the circle's kind of shifted around. Once ATP bound, it shifts the myosin into a low affinity state, disconnects it, and then you have dissociation and hydrolysis, and then once a hydrolysis occurs, you go back to the high affinity state, you bind, and so forth. So same story, it's a little different picture. But what I wanted to point out is, to get out of the high affinity state, you have to add ATP. So if you run out of ATP, you're locked. The device is locked to the actin. We call that rigor mortis. Some of you that have been in your previous lives worked in the cemetery business or the mortuary business and had to pick up cadavers knows that shortly after people die, they go into this rigor state. Maybe you're most familiar with it with picking up roadkill for dinner on the way home, but when animals die, they go through this state immediately thereafter that they become almost like a board, and that's this state. They run out of ATP and their muscles are all locked up. And it lasts until enzymes start chewing up the proteins and so forth. So anyway, that's the genesis, the cause of rigor mortis. Okay, so what controls this whole process? What causes this process to be able to be turned on and off? All we've shown you is a little cycle that keeps going on and shortening the sarcomere, decreasing the width. So what controls it? We call the control EC coupling for excitation-contraction coupling. And this whole story is kind of a cool story. One of those stories that, again, it would be nice to go over the F house and explain it to the lino, but this EC coupling is like 20 steps, and if you can explain it to the lino and to the ale house, you'll pretty well understand it. So that's about my level, so let me see if I can do it. So every fiber has what we call neuromuscular junction. And skeletal muscle cells, they have to have a motor neuron coupled them to contract. So each individual muscle cell or fiber has a nerve, which we call a motor neuron, going to it. And the junction of the nerve with the fiber we call the neuromuscular junction or motor neural junction. I've heard that term used too. It's a class of structures called a synapse. Synapses are connections between nerves and also between nerves and muscles that allows continuity of excitation, as we'll talk about in a very short time. And so let's say, and you don't know much yet because it's a big circular story what an action potential is, but there's a signal coming down the nerve, which we'll call an action potential, and it comes down, it goes through this motor end plate or neuromuscular junction and activates the same action potential in the sarcolemma. So we're allowed the same stereotyped change of potential that goes down the nervous cell, goes across that synapse and also activates the muscle membrane, as we'll talk about, which we call a sarcolemma. Now, what does it do once it gets to the muscle membrane? So this is another artist's depiction of a muscle, and this in orange around the side is the sarcolemma. And so we've just ignited or sent an action potential to that sarcolemma, and now to see what happens to that cross-bridge cycle, we have to look at very interesting membrane architecture of skeletal muscle cells. The sarcolemma has holes in it, or is perforated as represented here. And these holes are openings to what we call transverse tubules. And the transverse tubules are gold here. They're in between the green lines. It's not too clear, but the holes go into these transverse tubules, so they're openings into the tubules. The tubules literally go right through the whole cell. The T-tubules go, we call them transverse. They go perpendicular to the sarcolemma right through the cells. And there's one of these at each junction, and that's not represented. Oh, I guess you can see it over here. So at the AI junction, there's a T-tubule each time. So there's two tubules for each sarcomere. That's not true in cardiac muscle. It's kind of interesting. They're on the Z lines. But in skeletal muscle, there's two T-tubules per sarcomere. And you'll notice that they loop right around these myofibrils. So the T-tubules go all through, kind of forming a network around the myofibrils. Another characteristic of T-tubules is the fluid within them is the same constituency as extracellular fluid, and that's critical to their function. So those pores through the sarcolemma are not gated, they're just open holes, and the same constituency of ions that's present out here in the interstitial fluid goes right through the T-tubule, and so it wouldn't, if it didn't, it wouldn't work. So they contain extracellular fluid. So we have, like we do in Confident, we have little canals going between our homes, and so this is like canals going in between the myofibrils containing extracellular fluid. They can launch boats in them. Okay. Now, the green network here is another whole network of membranes, and it's called the sarcoplasmic reticulum. You heard about the endoplasmic reticulum when you talked about cells before. Well, this is just kind of a branch of endoplasmic reticulum. It's sometimes called a fenestrated bag. Fenestrated means has windows, and it looks like it has little holes or windows in it right here. And so some sort of artwork, window decoration. But what it is it's each of these, extending from this larger cylinder to this one here, is one continuous sarcoplasmic reticulum, and then there's these little trails in between that are also open. So this is a network of canals, if you will. There's a large one on each end, and then these little interconnecting tubules, if you will. So we call it a membrane enclosed vesicle, but it has a very complex shape. These lateral ends, if you will, or these ends right here on each side of the SR, are called cisterns. Cisterns hold water. They're things which we use to hold things. And so these actually hold calcium. But these are little bags on each side of the T-tubule. In fact, so I just gave that away, but the SR is used to trap or to hold high concentrations of calcium. And one we first saw before we figured out all the structure was a triad. So a triad is a controlling group of membranes that control skeletal muscle activity. So there's a T-tubule, and on each side of the T-tubule is a cistern of the SR. So here's another artist's representation of the whole thing. So in this case, the blue lines are the T-tubules, and the tan are the SR network, and on each side of the SR is this cistern that is butted up against the T-tubule. And so I think this is from Boron and Volpe, but it's another representation of the membrane system. So this whole EC coupling business is controlling the intracellular calcium concentration that controls muscle contraction. And it turns out that we're just doing it for skeletal muscle, but the change that occurs in calcium concentration between 10 to the minus 7 and minus 5, 10 to the minus 7 is very, very low calcium concentration that occurs at rest. It goes up to about 10 to the minus 5 molar during contraction. So that fluctuating up and down is what's going to control skeletal muscle, but it also controls smooth muscle and cardiac muscle. But the way an EC coupling works is totally different in each of these. So it's a cool story, and the muscle works the same way by calcium concentration going up and down, but EC coupling is really different in each of these types because it has a different purpose. We're not going to get into details of the other types. We'll do it later, but right now I just want to emphasize that this is going to be the story all the way through in different muscle families. So here's another depiction, if you can follow me. This is the sarcolemma and the T-tubule going into the cell, and here's the SR with the cistern on each side. And again, you notice this intimate connection, if you will, between the T-tubule and the cistern of the SR. And I already said that at the neuromuscular junction, action potential goes from the nerve to the muscle, and it runs along the sarcolemma. Well, because the T-tubule has an extracellular fluid on one side and intracellular on the other, it can conduct action potential just like nerve cells and muscles or nerve cells do. And so it can not only go along the outside, but it goes right down into the T-tubule. So the T-tubule allows us to get action potentials right through the depth of the muscle, so you get a really, really fast response. So the action potential goes down into the T-tubule, and it activates voltage-gated calcium channels. And so in the sarcolemma itself, there are, I haven't talked about this, and I'll talk about it very shortly, kind of indicated one time about membrane, but there are channels that conduct calcium through the membrane, and they're controlled by threshold voltages. And this particular calcium chain voltage gauge channel is called the L channel. It opens when the membrane depolarizes. And when that does, calcium goes in, and it will end up activating ryanodine receptors. And the ryanodine system is part of the SR. And so that will release the calcium stored in the SR. And so that elevates calcium. We've got a better figure coming up that I want to get more detail. But here's the action potential going down the membrane. It goes into the T-tubule. The ryanodine receptor, the pyridine receptor, the L channel opens, calcium goes up. Then you end up having the calcium released from the SR. The calcium binds to troponin. You can actually measure that. When all the troponin sites are bound to calcium, then the force occurs. So it's the calcium that's the controlling force, the controlling element to cause muscle contraction. This is a really cool picture that's in the boron and bulpate text that goes through this thing. So here is the T-tubule here in the center, and this is the L-type channel, or dihydropyridine channel. It's called the DHP channel because it's inhibited by DHP. But anyway, so when the voltage goes down to an action potential, that opens these channels, and calcium can go, in this case, from the extracellular fluid. Remember, the T-tubule has extracellular fluid, into the interstitials, or into the intercellular space of the muscle. This is sarcoplasm out here. It's thought that this alone, it may facilitate the opening of the ryanodine receptor that's represented in purple. And that's what it says here. Depolarization, calcium channel causes more conductance. But what the latest is that it's not that elevated calcium as much as a mechanical connection of these dihydropyridine receptors to the ryanodine receptor. And you notice the foot processes of the ryanodine. This is a ryanodine receptor, and they think mechanically the change in configuration of the L channel actually opens these ryanodine receptors, and they've done a lot of work on this. A lot of this started at the University of Miami. But anyway, they open these channels, and then this high amount of calcium that's stored in the SR is released, so calcium rushes out into the cell and binds to troponin and on and on. And this shows, I just wanted to go into a little bit more detail about what calcium does. So I said it bound to the high affinity sites on the TN, on the troponin C. So it binds to a pair, low affinity, this is right. This is right. The high affinity already bound. It binds to the low affinity calcium sites, and then you'll see the shift that occurs. The troponin I clears the myosin binding site, kind of rolls. It's like tropomyosin rolls over into the groove, and then that exposes the binding site on actin for the myosin head, and so the myosin head can bind to the actin, and then once it does, that starts the cycling occurring, the cross-bridge cycle. That cycle continues until the ATP is removed. As long as you the calcium is removed. I didn't mean to say ATP is removed. The cycling occurs until you decrease the calcium concentration. So the action potential causes calcium to be released. Calcium initiates contraction. The contraction will occur until the SR retakes up the calcium. So the sarcoplasmic reticulum is always trapping calcium, we call it. And once it lowers the calcium back to the 10 to the minus 7 molar, then the contraction will cease. And we'll talk about that a little bit more. But first, just introduce some mechanics of the process, and we'll get back into this in the next lecture. You can divide the muscle mechanics into two broad areas. One's called isotonic and one's called isometric. Isotonic means same tone, and isometric means same length. So this on the left here represents an isotonic contraction. So you have a muscle with which holding a weight, you stimulate it, and it lifts the weight. So the weight stays the same the whole thing, and what you're going to measure is a change in length with time. So we call it displacement. So if you're monitoring this contraction, you'll watch the muscle shorten and then lengthen again. So we monitor the change in length with time. Isometric contractions are keeping the muscle fixed in position and stimulating it contract, and it'll exert more force. So here you're going to measure the amount of tension and how it changes with time. So one's called isometric and the other's isotonic. Isometric type of muscle activity is when you work against a fixed object. So if you're a weight trainer, you've probably been introduced to isotonic training and isometric training. They're both mechanical mechanisms that muscles work by. So I have an clicker question on isometric versus isotonic. If you're walking from place to place, is that isotonic activity or isometric activity? Sometimes it's nice to actually think about applying these concepts. So when I'm constantly pacing the floor, is that isotonic muscle contraction or isometric muscle contraction? Go at about 1:30, so another 5 seconds or so. We got to 1:38. Okay, 5, 4, okay, that's good enough. I stopped at that time. Most people thought it was both. When you're moving a muscle, the muscle shortens, so the legs are indeed, as they shorten, exerting isotonic type contractile activity, but indeed other muscles in your body have to hold you in a given position. Also, you have antagonistic muscles, some of which have to maintain tone that are isotonic. So you actually have all kinds of both. And it's very complex. It's only really in an experimental model we can separate all the components. But it's useful, and you'll see it's extremely useful when you're talking about how the heart works. A little bit more on mechanics before I talk about the test. I mentioned before that each individual cell has a motor neuron going to it that causes it to contract. So all muscle fibers have to have one neuromuscular junction to cause them to activate. This is skeletal muscle, not smooth muscle or cardiac muscle. All skeletal muscle cells have a neuromuscular junction, but many cells can be hooked to the same motor neuron, and that's called a motor unit. So a single motor neuron, like represented here, can come out and not hook to one cell, but to many cells. And all of the muscle fibers that hook to a single motor neuron are called a motor unit. And that size is variable. If you had a really big motor unit, it would have a lot of power, sort of like having 7 horses hooked to a single bridle. Where one horse might be able to be controlled easier, 7 horses would pull you better. So the greater the size, that little symbol is proportional to. So the greater the size of the motor unit, the more force that muscle can produce. But it's also at the sacrifice of dexterity. If you really want fine control, you'd want a very small motor unit. So we have a lot of variants in motor units. Some of them have hundreds of cells, some of them have 4 or 5 cells. And so that leads to my next eye clicker question. Which of the following muscles would have the smallest motor unit? Oh, geez. I'm thinking my brain's in another space today. Okay. I think I have just one more, so I'm almost done here. I'd like to get through most of it. Five more seconds or so. Okay, 5, 4, 3, 2, 1. 2 people are up there hanging somewhere. Or maybe they left, they got bored. Okay. Everybody said the eye muscle, superior rectus. And that's true. They have extremely small motor units, just a couple of cells. And that means we can move our eyes in very small arcs because we have minute control. What would have the largest motor unit in this family? The gluteus maximus is a huge muscle in your butt, Eric. I've seen some really fine gluteus, but that doesn't mean they have fine control. I mean, just did all this work. Although I've seen pretty good control there too. But anyway, the largest motor unit would be the gluteus. That we call a muscle twitch a muscle contraction to give a muscle to a single stimulation, and it's useful to characterize a muscle. Lots of physical qualities come from a study of a single twitch. For example, if you stimulate a muscle right where the red arrow is at time 0 in milliseconds, it's going to be a while before it contracts, and that period is called the latent period. Latent means sleep. And that's when all those events which we've been talking about, excitation, contraction, coupling, take place. So that's the latent period, and it's hugely different in different muscle families. Then there's a period which it's shortened, and in skeletal muscle it's often about 20 milliseconds, 20, 30 milliseconds. And then you have the period of relaxation. So that's a classic muscle twitch and what it looks like as far as a graphical representation. And skeletal muscle twitches last about 1/5 of a second, 20% of a second, from 100 to 200 milliseconds. Another characteristic is this type of stimuli that initiates contraction. So this is the stimuli of a motor neuron, and you see this is amplitude. The height of this representation is the amplitude of the voltage. So let's say this is a nerve, and you stimulate a nerve with this much voltage, you don't get any activity, contractile activity. If you stimulate it more, you still don't get any contractile activity. If you get up to number 3, you get some contractile activity. We call that a threshold. So 1 and 2 are called subthreshold stimuli because they don't cause the muscle to contract. 3 is the threshold. So the threshold stimulation is the least amount of voltage that will cause contraction. If you keep increasing the amplitude of the stimulus, you'll get to a situation where all the cells are active within the given muscle, and that's called maximal voltage. So right there would be maximal, and once you go above that, it's supermaximal. No matter how much voltage you give, you've already got the muscle totally contracted, you won't get any force. This figure in the middle is very important, and you might just glance at it, but you see there's little neurons, motor neurons within that nerve lighting up in red. A motor nerve may have many, many axons in it. And what happens when you increase the voltage is you're activating more of the axons in a given motor neuron, which means that if you have the femur nerve going to the muscle, if you give a little bit of voltage, you get a couple of motor neurons through that nerve activated, but eventually you get them all and you get the maximum force, and that's what they're trying to show you, how many nerves are activated. This whole process is called spatial summation. In other words, when you increase the amplitude of stimulation to the motor nerve, you're bringing in more and more motor units, and so you're summating motor units. So once again, a typical muscle has, or nerve goes to hundreds to even thousands or muscle nerve goes to thousands of motor neurons. And if you increase the strength, you're going to bring in more and more motor neurons and activate more and more motor units and get more strength. So that whole process is called spatial innervation. So a couple quick eye clicker questions, then I'll talk about the test. Oh, I got turned on. Oh, that was interesting. I didn't do that. Try again. Okay, it's not started? I guess it won't do this when it doesn't like me anymore. There we go. Had to hit it 6 times. This is an important question. See if you're following, because this is, if you don't get this question right, you didn't follow that last year's story. Okay, another second or two. See, I just want to see how well you followed that. So, come on, machine. Ah, good. You really followed it. Okay, if you have an accident going down a nerve through a motor neuron through the sarcoplasmic reticulum, the single action potential will always have the same amount of force as skeletal muscle. In other words, if you increase the amplitude of that one nerve to that one neuromuscular junction, you're not going to get more force. It's just one twitch. And we don't modify force by this, and I'll get more into this later. So the increase in force when you stimulate it with greater and greater strength is you're increasing active motor units. Once you activate a given motor unit, every cell within that motor unit is producing all the force it can. It's not going to get more calcium or whatever. You're getting the more motor units activated. So as you increase to that nerve, more and more motor neurons going to each its motor unit, you're getting more and more force. So think about this. This is an important point, and we'll get back to it later. Maybe that's the next question. I'll talk about this some more next time, but is this how we regulate force? Is this important to regulate force? When I want to lift a pencil or lift a brick, how do I regulate how much force the muscle produces? Is it this mechanism? Oh, jeez. I did sleep last night. There we go. This will be interesting to see what you think. So how do I figure out how much force the muscle is going to produce? See what you think here. Couple more seconds. Okay, you're right. It looks like you followed that point. You said it's very important, which it is. In other words, it's an amazing computer. If I wanted to lift something heavy, what my computer says is you better activate every motor unit in that muscle to lift it up. And if I wanted to lift up a pencil, I only use maybe 5% of the motor units in a given muscle. So, and sometimes we get fooled. We're helping somebody move, and they say, "Get me that box," and you think that's the one with the books, and just so your computer says, "I can activate everything," and that's you go like that. In other words, your brain prefigures how much heavy things are and figures out how many motor units to activate. So it's a pretty amazing system, but this is very important on how we regulate muscle. Let me talk about the test real fast, and then I'll let you go. Oh, that was it. Never mind. I stopped there for a second. Okay. Okay. You guys did very, very well. The average on the first, last year I think was one of the best classes I've ever had. The average on